My name is Chloe Chua. Today, my team and I will be talking about how certain Bacillus species can be used to restore oil paintings. My team includes Rajeshwari, Jasmine, Lakshman, and Li Weishin. Without further ado, let's get into it. While science has taken humanity far in the past few decades, from the Mariana Trench all the way to the moon, today we're back on Earth. We're going to be looking at how some of the smallest life forms can impact the art world. Now, art preservation is a delicate process. Ideal conditions are required to prolong an artwork's lifespan. Usually, the goal would be to create a sterile environment to prevent further damages to the paintings. But recent advancements have shown us that it might be more beneficial to introduce microorganisms instead to help with preservation. Let's step back a bit. Why are the paintings degrading in the first place? Well, just like our bodies, oil paintings are home to a community of microorganisms. In 2012, an earthquake struck the Basilica of Santa Maria, where a painting hung on the ceiling, Incoronazione della Vergine, was damaged and desperate for restoration. Dr. Elisabetta Caselli and her team of researchers took on the challenge to restore the 400-year-old painting. To do that, the researchers sorted various parts of the artwork and grew the microorganisms present. They believed that there were microorganisms growing, feeding off the nutrients in the components of the paint, mostly the yellow and red legs as they were the most nutritious. A detailed analysis of the microbial and fungal communities dwelling in the artworks and involved in their deterioration are essential for the adoption of optimal preservation and conservation strategies. Once Caselli's team identified what kind of microbiome was present in the painting, they were able to create a probiotic cocktail containing 3 strands of Bacillus bacteria. The results had shown incredible suppression of the Bacillus bacteria, but nevertheless, further testing will be needed to ensure that the treatment will not be harmful to the painting itself. Caselli's work provides invaluable insight to what the future of art preservation could be. The biodeterioration process involves every type of cultural heritage item: monuments, stoneworks, easel paintings. Research in the field of restoration has paved the way for many innovative biological approaches, known as BioRestoration, where microorganisms are not only considered as eventual dangers for artworks, but rather as potential tools for restoration. While pigment-eating microbes may degrade priceless paintings, other microbes may just help us save them. Now I'm going to pass the time to Li Weishin to further discuss the Bacillus microbe.
Hello, I am Weishin, and today I'll be introducing to you the characteristics of Bacillus. So to classify the organism, taxonomy is usually used. So for the domain of the Bacillus is bacteria, the phylum is Firmicute, class is Bacilli, order is Bacillus, family is Bacilliaceae, and the last one, genus, is Bacillus. Structures and characteristics. Structure-wise, Bacillus is shaped like a rod and can form endospores through aerobic spore bearers. These bacteria are usually saprophytes, which basically means they are able to live in dead or decaying organic matter. Usually they are gram-positive, but in some rare cases they actually turn gram-negative when they grow older. And in terms of characteristics, during pathogenesis, Bacillus makes enzymes and aggressins. And aggressins basically means that there are substances that contribute to the virulence of pathogenic bacteria by paralyzing the host defense mechanism and using the host cell to reproduce. So the main way this bacteria infects is through enterotoxin, which is a substance that actually harms your digestive system, or hemolysis. And hemolysis basically is a lipid or protein that actually causes your red blood cells to burst. And this usually causes food poisoning in humans. And moreover, they are able to survive in harsh environments such as temperatures, salt concentrations, and pH. Usually the bacteria needs oxygen for sporulation, and the process also needs other components such as temperature, pH, and certain compounds. For the roles on Earth, Bacillus usually leads the cause of anthrax, which is a disease in humans. And this disease can be transmitted through ingestion, inhaling, or through direct contact of the bacteria on the skin. This bacteria also acts as a symbiote, specifically the spore formers, which they coexist on pollen, larval phases, and inside the alimentary canal of insects.
My name is Rajeshwari. Now I will explain what role Bacillus bacteria play in oil paintings and how they work. Bacillus bacteria in oil paintings have an essential function in preventing and controlling the deterioration of oil paintings. To alter the appearance of oil paintings, BioRestoration, an organic chemical has been used. BioRestoration relies on enzymes like lipases, proteases, and carbohydrates as well as bacteria that may perform a variety of enzymatic activities such as BCB, SRB, NRB to avoid the bioreduction in artworks. Following this, Bacillus bacteria in oil paintings can be used in the bioremediation process to protect the historical artworks, monuments, or structures from biodeteriorating bacteria and fungi colonizations through independent cultural research. In a restorative agent, specific Bacillus strains are utilized to make a biocompound solution. As a result, oil paintings may be kept clean and free from microbiological contamination. Currently, the parasitic and hostile nature of bacteria and the results of secondary metabolisms like green biocides have led to the current use of live bacteria and metabolites of create inorganic chemicals of vital relevance. Bacillus anthracis and cereus are considered as safe for human health. In the end, the germination of spores from vegetative bacteria occurs when the cleaning agent exposes surfaces. As previously reported, Bacillus-based biocompounds have been used to treat individual bacterial and fungal species isolated from an ancient easel painting, and the results obtained by Kessler's team suggest the possibility of using these Bacillus strains to decontaminate artworks, surfaces, and prevent the growth of contaminate isolates, bacteria, and mycetes in the future. Now we move on to the mechanisms. The mechanisms for bioremediation with Bacillus bacteria, analysis of the bacterial and fungal populations in the material structure was carried out from optical and scanning electron microscopy techniques. Bacillus substils, Pumulus, and the Megatherium were selected for use in the biocompound after the researchers had determined the microbiological variety of the microbes in the environment. The Bacillus species has attracted the greatest attention because of its wide variety of secondary metabolites with biological activity. Some strains of Bacillus substils, for an example, can produce antifungal peptides, and the bioactive peptides include with antifungal peptides, lipopeptides, and antimicrobial peptides. The role of microbial lipopeptides is to protect microorganisms against stressful circumstances such as sporulation. Biodegraded mural paintings were used to test multiple Bacillus strains against Cladosporium, Penicillium, and Fusarium oxysporum. The Bacillus strains produce biosurfactant lipopeptides that inhibit with fungal growth. Direct antibiotics in which enterogenetic strains create antimicrobial compounds to suppress or kill prospective pathogens is observed in the strains. To summarize, these Bacillus strains have demonstrated excellent antimicrobial activity through enzymatic activity, competitive entherogenisms, and the formation of bacteriogens and other bioactive substances.
Hello, my name is Jasmine and I will be diving into the outcome of interaction between Bacillus in restoration of oil paintings. A test was conducted by Dr. Elisabetta Casilla and her team, where the bacteria and molds isolated from the painting were seeded on selected agar media in the presence or absence of the biocompound, which contained spores of Bacillus subtilis, Bacillus pumilus, and Bacillus megatherium. When conducting in vitro assays to detect decontaminating activity of the biocompound, the antimicrobial activity of the Bacillus species had been reported to show the capability of Bacillus strains to counteract different types of pathogenic bacteria. The figure below shows the results that Casilla's team had obtained when the biocompound was spread. It shows successful inhibition of contaminating microorganisms after 5 days of incubation. The results indicated that the presence of Bacillus almost totally inhibited the development of all the tested microbes isolated from the painting. This suggests a non-specific inhibitory mechanism, mostly due to competitive antagonism, that has already been observed in other environments. This means that the biocompound has the potential ability to counteract in situ the growth of contaminating microorganisms that are potentially dangerous to the painting, suggesting the potential use of these microorganisms to prevent biodeterioration of artworks. As you can see in the quote below, the study says, the demonstration of the antimicrobial action of Bacillus bacteria against the microorganisms isolated from the painting indicates their potential for use in bioremediation. Well, it's not surprising since Bacillus spores have a long history of safe use in humans and are successfully utilized as antifungals in agriculture and as infection prevention agents in aquaculture and zoo techniques. Now it takes strides in the field of cultural heritage conservation. Now we'll move on to Lakshman discussing the impacts of this discovery on humanity and the planet.
So hey guys, my name is Lakshman Manasan, and in this segment I'm going to walk you guys through the impacts of which Bacillus will be used in restoring oil paintings, the impacts on both individuals and on the planet. So let's have a look, shall we? So right now I'm going to go through the impacts of safety on mankind, human beings, individuals like us. So it is essential to have a comprehensive understanding of the decay process in order to generate remediation solutions as safety measures. This will allow us to effectively eliminate and control development of microorganisms that are actively involved in the biodegradation process prior to the restoration and conservation process. Additionally, we need to build methods for preventative monitoring in order to ensure the longevity of intervention-based artwork protection. Because of this, we'll have a better chance of avoiding unfavorable outcomes, even when the cleaned artwork is exposed to different environmental conditions throughout time. Further studies has to be done to certain whether BioRestoration techniques are actually delicate or safe in the first place. This is something that needs to be determined over time. Biological technologies have significant potential as a strategy for preservation of culture and heritage due to eco-sustainability, ease of configuration and improvement, and the absence of risk to both humans and environmental health. So that's a plus for us humans and the planet, because we don't have to go through any troubles of pollutions or anything. Additional studies is necessary to monitor the efficiency and safety of the strategy, which includes the research that looks at both short-term and long-term effects of using this method. Monitoring the biological activity and minimizing the unintended side effects of biorestoration procedures requires a prompt and consistent testing, such as measuring the amount of ATP present in the activity of the dehydrogenase. The demonstration that these novel technologies are free of known dangers will stimulate their wide adaptations, which in turn will lead to the creation of standardized protocols and procedures which have cost-benefits evaluations. By further developing bacteria for the purpose of repairing paintings, we can get to understand how the safety works on the safety is placed on human beings and the planet. So to my belief that if we put more effort into understanding how this technology works, we can prevent a lot of harms in the future. So moving on to the next impact. So the last impact that I'm going to talk about is how we as humans, individuals, can get a better understanding of our origins. So by further developing bacteria for the purpose of repairing paintings, humans have the potential to conserve thousands of historical works of art. This is because any future degradation of chemicals or materials will be both preventative and reversible. This will help people in the process to further restoring old artifacts and relics, which in turn may help us to have a better understanding and knowledge of where people came from as a species. Our most potent instrument to have a better communication with someone from another century, another time era, another culture will be improved and enhanced. And it will also allow future generations to enjoy and understand the importance of human heritage, history, and even our own legacy. Well, that's all from me, and I really hope that everyone was able to take something away from this presentation and add it to their interests. I really hope that oil painting will continue to be as successful and as effective as it always has been, because I can't wait to see the Mona Lisa when I grow older. It's very much appreciated that you stayed until the end, and thank you very much for hearing us out. I'm signing out. Thank you. Goodbye.
Here are the references for our video, and thanks for watching.